How has the community been reacting to your research? It seems like it kind of flies in the face of everything that we've been told, which is that the only possible intervention is, progressively harsher and harsher chemotherapeutic agents.
Yeah, it's depends on who you ask. Uh, it's not universal in the response to our stuff. If you ask oncologists who take care of folks every day, um, they say it's all nonsense because they never heard about it. If you ask scientists, they say, yeah, well, we know mitochondria have problems, but we still think it's a genetic disease. Otherwise, I won't get my grant from the National Institute to Health. Um, when you talk to folks, they seem to want to know more about this. I haven't spoken to people in the pharmaceutical industry. I don't know what they think. They more or less do whatever the boss tells them to do. I find that's mostly what happens. My colleagues uh, think they're locked into confirmation bias, and I think a lot of folks are that you can't buck a system that's been in place for so many decades, even if you have accurate science. So I don't know how you respond to that. Um, it can't be right because everybody else thinks differently. So I don't think anybody wants to have the apple cart turned over. I think everybody wants to keep the status quo, regardless of what field or occupation or discipline you're in. I think the people, of course, would like something different.
I was going to say, like, except for the people with brain cancer, they probably would
But
like to
not
see the
only
apple
not
cart.
only brain cancer, all cancers. I get hundreds of emails and from everybody, every type of a cancer, and they all want metabolic therapy. But the institution, the system, is not allowing it for a variety of reasons. And none of it has to do with patient health.
Okay, well, maybe we can set the stage a little bit. So, you came to our attention because you kind of had a blockbuster paper that just came out about glioblastoma. Glioblastoma is a notably difficult cancer. Shiloh studied it for many years, and fatality rates 5 years out are almost 100% with traditional treatments. And yet, with this metabolic therapy, in conjunction to the standard of care, which is chemotherapy, you see 3, 4 years out people that are cancer free. Like
Not
unheard
only
of?
that, when I was studying it at least, and I haven't been in the field in over a decade, but it seemed like they hadn't really made progress with the standard of care in more than 20 years at that point. And so that was really shocking. Plus, the standard of care, I think, improved overall survival by a matter of months, right? So it was a really, I actually left that field because it was so depressing. I was just like, God, what are we doing here? Um, so it's one of those diseases that's generally taken as a death sentence.
Yeah, exactly. And this paper seems to suggest that there's an alternative that's possible.
Yes, that's true. Um, it hasn't. There's been no progress in 100 years, not 20 years. If you go back and look at the data from Bailey and Cushing in 1926, they said that people with glioblastoma uh, and very aggressive brain tumors um, would survive anywhere from eight to 14 months, which is today it's about 8 to 16 months. So that's you got an extra couple 3 months in 100 years. Um, so you're right. It is, but it doesn't have to be. It doesn't have to be. This is the great tragedy. We can manage that. We think we can make 4 years the average survival rather than 15 or 16 months, maybe even longer. I wrote a paper in 2010 in Lancet Oncology. We were just so upset about the tragic loss of lives with this disease, GBM, of course, that I just fired it out and I said, let them gnaw on that. And we clearly said that the standard of care is responsible for the demise of these people. Um, because as a neurochemist, I understand the glutamine-glutamate cycle. And we know in the brain, glutamine is a very necessary amino acid for the synthesis of glutamate, which is an excitatory neurotransmitter. And it has to be removed very quickly after being released, that is, the glutamate, because it impacts on NMDA receptors in the neighboring neuron to transmit signals from one cell to the next. And if it's not removed quickly, it can lead to excitatory seizures and excitotoxic death of cells. So we knew this. And we knew that as soon as the glutamate is released into the synaptic cleft, the glial cells, which embrace the synaptic cleft, would take up glutamate very rapidly and metabolize the glutamate to glutamine, which is not excitatory. And the glutamine would then be released by the glial cell and taken up by the neuron. And the neuron would then metabolize the glutamine back into glutamate and consolidation in synaptic vesicles. And this has been studied in the neurochemistry field for decades. It's called the glutamine-glutamate cycle, and it protects the brain and neurons from excitotoxic death. There had been some reports that in glioblastoma, there was a significant amount of extracellular glutamate, which was leading to excitotoxic death of neurons just in the microenvironment. And I knew that there's plenty of astrocytes that would be sucking this up. But then we also realized that the tumor cells themselves depend heavily on glutamine. So if the glutamate is being released from the neuron, or the neuron dies and a significant amount of glutamate is now in the microenvironment, the astrocytes will suck up this glutamate and then dump it out as glutamine. And if there are cancer cells in the microenvironment, they're going to be sucking down the glutamine. And we now, at that time, in 2010, we knew that debulking surgery followed by radiation. Radiation heats the brain and would lead to significant neuronal death, more glutamine cycle disrupted. Any surviving tumor cells in the microenvironment would be bathed in a fuel that they need to survive. And not only that, after your brain is irradiated, and right after surgery, and even before surgery sometimes, because the necrosis and inflammation and edema in the tumor microenvironment is treated by high-dose steroids. So we knew that the steroids would raise blood sugar levels. And not only that, when you irradiate somebody's brain, blood sugar goes through the roof. It's an attack on the body. So we have two fuels that the tumor cells absolutely have to have. The sugar, glucose, created by the radiation and the steroids. And then we have the glutamine being dumped out onto the microenvironment, leading to the survival of the tumor cells, making them massively resistant to any further attack by chemo or radiation. Because the waste products of glucose and glutamine, which we now know is lactic acid and succinic acid, protect the tumor cells from any further attack. So the reason why they recur and are resistant, because of the very treatments that these poor folks are receiving by the standard of care. It makes no damn sense. If you have any clue about neurochemistry, neurobiology, or cancer biology, what you're doing to your patients is essentially depriving them of any possibility for long-term survival. I have published this over and over again, detailing all of this, and it falls on deaf ears because the system is so profitable using radiation and these crazy things that it's not possible to shift gear regardless of what the patient, how it might affect the patient. This is immoral, absolutely immoral and ignorant on the part of the field. So you take patients and you don't do that, and they live longer. And they can live a hell of a lot longer if you didn't irradiate. And those papers, the paper you're talking about, because my Greek colleagues we're still forced to do that stupid radiation. And they know if we didn't irradiate those people, we could even get them to live far longer.
If you want to learn how to implement the ketogenic diet and the Press Pulse protocol into your cancer management, just visit school.com/keto for cancer, the new community by our research analyst Johnny Rockermeyer. Within this new community platform, you can connect with other patients and learn from their experiences. You can also just search through the posts other members have already made. You will have access to our classroom section as well, which provides practical advice and tools for the best results. These sections cover all parts of the Press Pulse protocol that you should implement. Starting with the general scientific concepts by Dr. Thomas Seyfried, we will show you how to start the ketogenic diet, including which foods to choose and which to avoid. Additionally, we prepared a list of doctors specialized in metabolic therapy, as well as details about glutamine inhibition and hyperbaric oxygen protocols. So, take a look around at school.com/keto for cancer.
So
Yeah, I was going to say, isn't that a piece of the ethical guidelines for designing these studies is that you have to maintain the standard of care when comparing new therapies? Is there something about that I recall?
Yeah. Well, the system locks in. It prevents alternative views to a standard of care. They'll say, like at Cedars-Sinai in Los Angeles, you can treat without standard. You can treat with standard of care, but you can't do metabolic therapy unless you've already done standard of care. So you've already stacked the deck against the patient. So what are you going to do about that? That's your question.
Yeah. So were you using some sort of was it? Let's see. The tumors love the glutamine. Were you using an inhibitor of the glutamine in this trial? I don't recall that.
No, we did not. That's another thing that we could even advance. We could even improve the outcome even significantly more.
And what was the challenge to pulling that off? Why couldn't you include that?
Well, it's still not readily available. Again, this was done before some of those patients were being treated years ago, and it was before the big treatment protocol that we just wrote at the end of 2024. We've written a very comprehensive treatment protocol, which says don't irradiate the brain, basically. So, and we gave many options of how you negotiate the treatment strategy depending on the individual. One of the authors on our papers is Dr. Joseph Maroon, former president of the Neurosurgical Society of America, and he told me he operated on 400 GBM patients on his life, not one survived. So he said we got to do something. So we know, and I gave a lecture to Henry Ford Cancer Center, to the radiation oncologists, their whole division of radiation oncology. How do you think that went over when I told them you were killing all your patients? That didn't go over well. You might as well have been, all I did was get glares, rather than saying, well, I guess it was pretty harsh to tell somebody that their whole life is just like, what are you doing?
Well, it's crazy, right? Because we've convinced ourselves that our medical interventions are a lot better than anything that was done in the past. And we're working on a book right now, and I was looking through the Ebers Papyrus, which was this medical text that was unearthed from 1500 BC in Egypt. And there's some stuff in there that's kind of relevant to what we do now, which is like various, like belladonna for seizures. You can make a couple of jumps and say, well, it is a sedative and it does seem to help in epilepsy. Guinea worm, we still treat the exact same way that we used to treat. It's like a 3-foot long worm that the only way to get out of the body is to just like slowly wind it around a stick until it comes out. Like we haven't figured anything better out. But then there's these really crazy things where they're like, okay, so if you're blind, you have to take a crocodile and a clay crocodile effigy, stuff its mouth with herbs, and then tie it to your head with a piece of linen that has the names of God inscribed in it. Or you have to do, if for other eye problems, you have to take like the dust of granite and apply it as a poultice to the eyes. And you look at it and you're like, this is insane. But they did it for hundreds, if not thousands of years without any real reflection on whether or not it works. And we pat ourselves on the back all the time about the fact that we have this really evidence-based medicine.
Hmm.
And if we've been doing the same thing for glio for 100 years and there's not any improvement, it seems almost incomprehensible. Like, how are people not looking at the standard of care and at this conference just overwhelmed with relief at the fact that somebody's offering them some option that improves outcomes?
Yeah, it's remarkable. You'd think they would be excited about it. But they're threatened or feel threatened. And it's really a very unusual situation. But it's not, when you go back and look at massive paradigm changes from the past, you find the same kind of reaction by the establishment. This is what I'm experiencing here is no different than what Galileo had to deal with, and Giordano Bruno and Kepler and these other guys when they were defining the sun as the center of the solar system. I mean, this threatened the supremacy of religious organizations and things like this. So and it takes decades sometimes to accept even what is obvious. And we still have 15% of the American public still thinks the sun revolves around the earth. So we've made a lot of advance, but not complete. Um, and you can say the same thing for Louis Pasteur. He was brutalized, even though he came up with a rabies vaccine and found out that it was germs that was causing infectious diseases. And the French hierarchy belittled him. And we still have the same problem with the Darwin-Wallace theory of evolution, which you cannot understand glioblastoma or any cancer effectively unless you understand evolutionary biology. It's not possible. So if you have people that don't understand evolutionary biology, they're the same people that would put the alligator stuffed with herbs in your face. You know, so you
I'd
know,
like to believe that the country's leading oncologists have at least like a rudimentary
Well, I think
grasp.
no,
he's not pointing
no,
at the oncologists so much as the technicians, like the technical radiation oncologists. Like they are oncologists, but their job is to shoot people with beams of radiation. Like what are they gonna do if they don't get to shoot people with beams of radiation? They've spent their whole lives training to do that. It's like a whole cast of society that's out
Yeah,
of a
yeah,
job,
yeah.
and you
yeah.
can't teach an old dog new tricks, perhaps.
And not only that, they get very well paid for this. I mean, you're talking about these radiation oncologists get a lot of money. So now I'm not saying all radiation is bad. I think for brain it might be for sure,
Mm.
but I think for as a final removal of some lesion, a proton beam could be the solution to the problem. Um,
It does debulk the tumor significantly. Like the short-term gains are evident, right?
yeah, but if we take and again a glioblastoma and shrink it down and make it less angry, less inflamed, less angiogenic, where now the surgeon can debulk a far greater amount, and what's remaining may be susceptible to a low dose chemo or even these parasite medications that we have. But we would not, and we do not recommend radiation to the brain for glioblastoma or any brain cancer patient for that matter. But that does not mean we could not use radiation for lung cancer, colon cancer, or something like that. If the lesion remaining is tiny and compacted, because if you don't do that, you could run the risk of spreading it all over the place. And don't forget the reason why radiation it kills through reactive oxygen. Reactive oxygen is like a bomb, a local bomb blows up, kills the cell. But if you don't kill that cell, and that cell is fermenting glucose and glutamine, it's got kind of an acidified shield around it, making some of these treatments ineffective for killing off all the tumor cells. So you really have to put the tumor, regardless of where it is, because what we have found is that all tumors, regardless of where they're localized, have the same metabolic problem. They are powerfully fermenting glucose and glutamine, dumping out large amounts of lactic acid and succinic acid, and cannot shift to ketone bodies or fatty acids to replace glucose and glutamine. So what I just told you is the solution to the cancer problem in a nutshell. Um, but that's not taught in any medical school. It's not, other than our papers, there's no one else saying this. Yet we have all the science to support what I'm saying. Based on historical observations from Otto Warburg, we have a big paper that'll be coming out, just accepted for publication in Bioenergetics and Biomembranes, which really tells the entire cancer field where we went wrong and how to get back on track to manage cancer effectively, because it is a mitochondrial metabolic disease. So the mitochondrial metabolic theory will replace the somatic mutation theory. And then the field can start moving forward with treatments that are far less toxic and far more therapeutic, because it's based on the science as we know it.
Okay, so let's pull on a thread. What were you going to say?
Well, I was going to say that it's probably not just the clinicians that are an obstacle to adoption. It seems like asking people to avoid sugar in their diets is a huge ask as well. What were you going to say? We can get back to that.
Well, I was going to say if we can pull on that thread of what Dr. Seyfried said earlier, which is that you have to understand evolutionary biology to understand this theory. And I want to know what you mean by that.
Yeah, so the cancer cell is simply falling back on its default energetic state, which is fermentation. All organisms on our planet began in an anoxic environment. There was no oxygen in the environment. So all the cells, the living cells at that time, were fermenters. There was no oxygen. They were all single cell fermenters, and they would grow, divide, and would live as long as there was an availability of fermentable fuels in the microenvironment. Then we had certain archaeobacteria that were able to produce oxygen. And over time, we developed oxygenated environment. It was the oxygenated environment that allowed us to form metazoans, which are multicellular organisms, because the fusion of the archaea with other fermenting organisms at that time now produced a hybrid system where one organism could use oxygen as an acceptor of electrons, and the other organism was linked to a fermentation metabolism.
And this is actually like held together in the evolutionary system today, because like a human cell, a eukaryote, has fermentation that occurs in the gooey part, the cytoplasm. And then the oxygen-based metabolism occurs inside of the mitochondria, which is supposed to be this organism that merged with the fermenter. This is what you're pointing to.
Yeah, and yes, and then now we have division of labor, and we have energy efficiency, which led to metazoans, which are multicellular organisms. Now we know from the work of Sonnen Shinisato, Carlos and Anna down here at Tufts Medical School, they have been professing for a long time that the proliferation is the default state of all metazoan cells. When no longer held in check, they will naturally fall back on proliferation. And what we have shown is that the default energetic state is fermentation. So what's keeping cells quiescent and differentiated is the action of the mitochondria, producing highly energy with using oxygen as an acceptor of electrons, leading to a highly energetic state, allowing cells to differentiate and form different organs and different systems, and can operate under different conditions. Well, the mitochondria energy gradient inside that organelle, which has its own DNA, as a matter of fact, relinquished all genes to the nucleus except 13 genes as part of the electron transport chain. So they essentially control the destiny of the cell. And when the cell gets sick, they release cytochrome C, and the cell dies to protect the rest of the cells from any dysregulated cell growth. So the organelle that controls the destiny of our existence is the mitochondria.
Hold on, can I stop you just for a second?
Yeah.
Can you elaborate on the role of cytochrome C? So cytochrome C is one of the oxygen acceptors that's in the electron transport chain. Is it protective in the sense that it just sops up all of the reactive oxygen species that are released?
No, it doesn't do that. That's glutathione. That's in the cytoplasm. You have the pentose phosphate pathway, and you have the glutathione system in the cytoplasm.
And so, what's the role of the cytochrome C? When
cytochrome C is called the respiratory enzyme. This is what Otto Warburg got the Nobel Prize for. He found out the protein that actually allowed cells to use oxygen, which is cytochrome C. So when you take cyanide, you block cytochrome C's ability to accept move protons, and you kill the organism real quick. So when cytochrome C is released from the mitochondrion, it's a signal to die. So you're not respiring anymore, and that's called a program cell death, apoptosis, program cell death. which means the mitochondria are damaged or inefficient in some way. Cytochrome C is released, sending off a signal to die. That's called programmed cell death.
And you're saying that's protective for the other cells that are involved just
Yeah,
because...
it keeps an organization when you're not able to function energetically any longer. There's a program that triggers your death.
So it's
So
basically protective for the function of whatever organ the cell is part of. So you don't start to accumulate dysfunctional liver cells or brain cells
yes,
or whatever. Okay,
yes,
that makes
yes,
sense.
and basically, when oxidative phosphorylation is chronically disrupted over time, which was Albert Szent-Györgyi oncogenic paradox. He wrote that in 1977 in the journal PNAS.
So one second. So for people that have a hard time following the biochemistry, oxidative phosphorylation is the process of regenerating ATP from ADP. So we have this molecule that travels around the cell that is used as energy exchange for for literally everything. Like if you read Nick Lane's work, he's
Mhm.
basically making the argument that we go through there's more power density in a human body over the course of a day than there is in the sun, because we're constantly cycling these molecules as we shuffle them through the cell, and we break a phosphate off of this molecule in order to make some kind of reaction go. That depleted molecule comes back into the mitochondria, and the mitochondria's function appears to be to recover this pool of molecules that are able to be exchanged for work elsewhere in the cell.
Yeah,
And so
and,
oxidative phosphorylation is the process of reproducing the pool of this ATP molecule that you need.
yes, absolutely, and every breath that we take, that's why we're breathing here. Every breath that we take brings oxygen into the system. Every exhale comes out, it's got CO2 and water moisture. So that's the waste product. The waste product of oxidative phosphorylation is CO2 and water. And the CO2 are the carbons from the foods that we ate, and these foods are broken down systematically in the Krebs cycle, and then we generate these reducing equivalents, which deliver their electrons to the proteins of the electron transport chain, which is the mitochondria, and the proteins are moved from one complex to the next, from one protein, and they pump protons into this inner membrane.
Sorry, you said proteins, but you're saying electrons. The electrons are
The,
passed from
no,
complex to complex.
the electrons are passed from protein complex to protein complex that's embedded in the inner membrane of the mitochondria. Okay, and there's lipids, and these lipids in the inner membrane are facilitators of how efficiently this whole thing will work, like cardiolipin, a unique lipid in the inner membrane, and the electrons are passed from one complex to another, and these complexes are multi-dimensional protein conglomerates. And in the process of moving the electrons, the protons are pumped from the matrix into this inner membrane to space to form a gradient, a high proton enriched gradient, which then pass through another protein complex called the F1F0 ATPase, and that spins like an impeller. And you're right, there's transporters that bring ADP into these mitochondria, and the phosphate is then added to ADP to form ATP, which is so highly efficient with the waste products of CO2 and water. So most of the energy in our cells is coming from this oxidative phosphorylation. Okay, so in cancer, that process becomes gradually corrupted, not acutely, because if you acutely disrupt OXPHOS, you kill the cell. And the example of that is cyanide. Look how fast you will die. That's called rapidly acute death, because you block cytochrome C, you block the movement of the electrons through the complex, and you block the ability of oxygen to be reduced to water. So this is basically how fast you can die. Now that does not happen in cancer. It's a gradual process. And when I go back to the oncogenic paradox of Albert Szent-Györgyi, he said something unique about cancer. It can be caused by almost anything we can think of, whether it's a virus, an inflammation, a radiation, a chemical carcinogen, intermittent hypoxia, viruses. He says, it seems like anything and everything and anything has the potential to cause cancer. But he was unfamiliar with the common pathophysiological mechanism, which allowed a cell to lose its growth control and become dysregulated in growth, which is cancer. Cancer is growth control is dysregulated cell growth or cell division out of control. So what is responsible for the control? Why cells don't do this? And the answer is because the mitochondria control the cell cycle. Through the proton motive gradient, they are controllers of the cyclins. They determine when a cell needs to divide or not divide. So they are the determiners of cell existence and destiny. And when
And
that
it's also
organelle,
worth pointing out that I think a lot of people have a model of the cell that they have from like an introductory biology textbook where
right?
there's maybe like one mitochondrion, but
Yeah.
there are hundreds of mitochondria per cell. They're moving around the cell. This is like an immense part of the cellular resources are
Yes. Yeah.
directed and put towards the maintenance and the survival of the mitochondria because they almost it's crazy because you can look at these videos of light microscopy where they've tagged the mitochondria and you can see they're actively moving around the cell. They seem to be organisms inside of cells
They are.
that are actively doing things still,
Yeah.
which is crazy.
It's a hybrid system. And my colleague, Dr. Shanopoulos, Christos Shanopoulos from Semmelweis University, is a world leader on mitochondrial substrate level phosphorylation, which we can get into. But he has that same video with, it's like a spaghetti network. It's like a bunch of worms in the cytoplasm. They are very active. The nucleus sits there as some, dormant thing, but it's the movement and the activity of the mitochondria. They fuse, they come together, or they break apart. They have their own DNA, but the DNA is in these little circles, and they have numbers of these different kinds of circles of DNA. They're a very active, dynamic organelle that leads to the entire energy balance and destiny of the cell. Ultimately, it's responsible to the second law of thermodynamics, which is disorder. When mitochondria become corrupted over age, they become gradually more and more less capable of maintaining energy balance, and you die. So you die because your mitochondria no longer are able to maintain a current across the membrane. So, and that goes back to the membrane. What's this current? Because you can't live if you don't have a current. It's called equilibrium, and equilibrium is death. So you need energy to prevent equilibrium. And every cell has a different electrochemical gradient across the membrane. For example, the heart has about a minus 88 or something like that, which means because the heart muscle is pumping. In order to do that, it has to have a very powerful sodium potassium ATPase and calcium ATPases, ATPase, breaking down ATP. You contrast that with an erythrocyte, which is a blood cell that carries oxygen around our body, picks up oxygen from the lung, delivers it to the tissues, picks up the CO2 from the tissues, delivers it back to the lung. Okay, that has a electrochemical gradient of minus 6 millivolts compared to the minus 88 millivolts of the cardiac. So interestingly enough, because the erythrocyte has no mitochondria and no nucleus, its job is simply to transport gases through the body, and then eventually it's recycled. But it still has an electrochemical gradient, a very low electrochemical gradient, and the energy for that organelle, for that cell, comes purely from fermentation because there is no oxidative phosphorylation. So they have a small fermentation, and they only need to produce a slight gradient enough to remove gases. Whereas cardiac myocytes and neurons, which have a very powerful role, have very steep electrochemical gradients, and therefore they have to have a lot more energy, and that energy is delivered by oxidative phosphorylation. Neurons and cardiomyocytes rarely, if ever, become cancer because they can't shift to a fermentation metabolism. So that's why you can know why these neurons never form cancer, or not never, but rarely, if ever. Cardiac myocytes, only cells that have the capability of transitioning from from oxidative phosphorylation to a fermentation metabolism have the ability to form dysregulated cell growth, all controlled by the mitochondria. So people need to appreciate more this importance of this organelle. Yet the whole cancer field was based on somatic mutations in the nucleus. So the focus of the field was on something other than the important issue, which was mitochondrial biology. And so yeah.
Does the oncogenic paradox come down to different means of injuring the mitochondria?
Yes.
Is that what you're suggesting?
Yes. Every one of those different injuries has a way to chronically disrupt oxidative phosphorylation, leading to what we call substrate level phosphorylation, which is the evolutionary conserved way that we had got energy before oxygen came onto the atmosphere 2.5 billion years ago. This was all single cell, all about fermentation. And the cells would continue to divide as long as they had fermentable fuels. Cancer cells are doing the exact same thing. The very organelle that's maintaining differentiated state and energy efficiency has lost that. So the kill switch, even though they can release cytochrome C, they're already locked into a fermentation metabolism, and they bypass programmed cell death, and they continue to grow in a dysregulated way, as long as they have the fermentable fuels in the environment to do that. Once you remove those fermentable fuels, these guys can't survive. And we interrogated them with all the different. We asked whatever amino acids, how many other sugar, what keep these alive? Our big paper just showed this. We interrogated all 20 amino acids. We interrogated everything and just asked what keeps these cells alive. And we were able to find it's glucose and glutamine, and they're powerfully synergistic. So when they get together, they will drive a dysregulated cell growth through a fermentation metabolism involving substrate level phosphorylation. Another big thing we found is that the damn mitochondria can ferment. There are fermenters too.
Wow.
There's a fermentation mechanism inside the mitochondria. It's called mitochondrial substrate level phosphorylation, driven by the amino acid glutamine. Glutamine is fermented inside the mitochondria. This is big. This is what was keeping everybody from understanding what the hell was going on in these cancer cells. It's unbelievable.
Hey,
Once you understand what I know.
What's up with the erythrocytes, right? Because they don't seem to develop cancers either.
No, they don't have a nucleus. You got to have a nucleus to divide. So if you don't have a nucleus, they're going to divide. So they don't have a mitochondria or a nucleus, and they can't divide. You have to have a cell that can divide. And what controls the division and regulation is the mitochondria. So a cell that can divide will proliferate like crazy. If it doesn't have a regulatory system to keep it in a stable state, and that's the mitochondria. That's why it's falling back on the default state, which is proliferation driven by a substrate level phosphorylation. And then listen, how do you prove that? Pull the plug on the glucose and glutamine and watch them die.
Well, one's easier than the other for everyday people, right? So we can control our diet, but glutamate is an essential amino acid. It's in almost all of our food, and so that's a little trickier, right? You know, I've had family members and friends going through cancer therapies, and I've talked to them a lot about your work. And the piece of the puzzle that's really tricky is the glutamate side of things.
Okay, I want you to repeat after me. There is no diet that can target glutamine. Can you guys say that?
I understand that. Yeah.
Okay.
Well, so this is actually really interesting because when we had this friend who had breast cancer, I started looking into it and I realized that the agents that are used to control glutamine and glutamate are basically chemotherapy agents, right? Because you have to block the proteins that are involved in this synthesis pathway, right?
Well, I don't know about that in regard to that. Well, but you're right. We can easily control glucose in our environment. It's a non-essential fuel. Our body can make it under restricted conditions through the process of gluconeogenesis. But the glutamine is an essential. They call it a non-essential amino acid because it can be produced from glucose by a glutamine synthetase reaction. The problem is for cancer, it's absolutely essential. And for our immune system, it's essential. And for our gut, it's essential. Urea cycle, it's needed. So you have an amino acid that's absolutely essential for our normal physiological functions. And it's the highest amino acid in our bloodstream. It's the most abundant. It's about 500 micromolar, 0.5 millimolar, and it's higher than all the other amino acids. And it's therefore an essential reason for the gut, for the immune system. If we get attacked by bacteria or a burn or something that leads bacteria into our body, our immune cells use glutamine as a fuel to kill bacteria and to perform its normal function. So do our gut needs glutamine, gut health. So sometimes you see people that are irradiated for cancer get high dose glutamine to repair their gut from being brutalized. So you have an amino acid that the cancer cell absolutely needs, and our immune system absolutely needs. So you have the same fuel that these tumor cells need to grow, and our immune system needs to kill the tumor cells, and they're both going to need the same fuel. That's why I developed the press pulse therapeutic strategy, where we press glucose down tight and exercise, because the exercise will pull the glucose out of the bloodstream. Our brain can switch to ketone bodies, but it will take what little glucose might be there. It leaves not much for the cancer cell. So now you come in with your strategic targeting of glutamine, but you do it in a strategic way. You don't do it in a non-strategic way. You have to know that your immune system is going to be needed. You've got to maintain gut health. You have to maintain immune system health. So, but here's the interesting thing: when you take away glutamine from the immune system, it doesn't kill them. It just paralyzes them. They can't perform their normal function. But when you take the glutamine away from the tumor cell, it kills them, as long as you put the noose around the glucose. So it's a balanced system. So what we do is we put the noose around glucose, and then we come in with a pulse of a glutamine inhibitor like 6-deoxynorlucine, DON, paralyzes the immune system but slaughters these tumor cells. Then you pull the drug out. You let the patient recover. The immune system then immediately becomes active, because they weren't killed, they were just paralyzed. So they come in and start picking up the dead corpses from the tumor cells and cleaning up the environment and protecting the gut. And then you come in and pulse glutamine, just glutamine targeting, just while you choke hold the glucose. And then the patient's tumor gradually gets degraded. And you don't suffer from hair loss and all the diarrhea, fatigue, and all this crazy shit that people do. Once you understand evolutionary biology and you understand the biology of the tumor, this is far less complicated than people have made it out to be. The problem, what I'm telling you, is unknown to the majority of people working in the cancer field. Even though we publish these papers in open access, peer-reviewed journals over and over and over and over again, they don't read it. They don't read the information.
Even worse, even if like when I tell, I literally know people who want to try these things, but they don't have access to these protocols, right? Like, how does somebody get involved in this glutamine pulsing protocol?
Yeah, well, that's what we wrote, the big treatment protocol with. We had 30 authors on this: MDs, PhDs, nutritionists, biochemists, physicians, the whole group. They set the standard. This will be the standard of care for cancer in the future. It should be today. We can save so many lives. We have 1,700 people a day in the United States dying from cancer. That's 70 people per hour, and these people are usually dying horrific deaths. They're not surviving the toxic treatments that they're given to these folks. Whether you have a brain tumor, colon cancer, lung cancer, breast tumor, they keep poisoning and irradiating these people using immunotherapies that sometimes work, but sometimes can kill you. And that's based. So you have to say, under what theory is your treatment being applied to me? And if they say, well, it's a genetic, some people don't even know. They don't even know what the hell theory they're working under. So if you're saying it's a genetic disease, then you're working under the somatic mutation theory. And our new paper that will be just accepted will be coming out in a few weeks. That clearly shows that the somatic mutation theory is no longer a credible theory to explain the origin or treatment of cancer. So once they come to know this, it will allow original thinkers to say, we got to go this direction. You either get on this direction or get the hell out of the way, because you're partially responsible for the death rate of these poor folks.
Is it going to be possible for somebody to take this piece of research to their oncologist and say, I want to try this protocol?
Yes, and guess what happens? Never heard of it. Can't be any good. No clinical trials. They throw it right back in your face over and over again. And then when the people do it themselves and they do really well, they come back to the oncologists, and the oncologist says, wow, this is unbelievable. I've never seen anything like this. I don't want to know what you're doing. Just keep doing what you're doing.
Well, they can't be doing the amino acid inhibitor, right?
Oh, sometimes they do. You can. What we found was we don't use DON, but we can use M-bendazole and fenbendazole and these kinds of things.
Is that some like over-the-counter business or something?
Yeah, dog dewormers and things like this.
Ah.
So why? So people say, why would you pull that out of your ass and do that? Because I did the damn research, for crying out loud. We found out that the embendazole parasite medicate. The parasites in the cancer cells use mitochondrial substrate level phosphorylation. The energy metabolism of a parasite and a tumor cell are very similar. But the parasite has a mitochondria that's not dysfunctional. The cancer cell has a dysfunctional mitochondria, but they're both using the same metabolic pathways. So parasite medications, especially under low glucose conditions, are you kidding me? Exercise, low glucose, take parasite medications, it destroys these tumors. Unbelievable. So, but I tell people this, I publish the articles, and they choose to ignore it. So what are you going to do? What, I mean, the people hear me. They go out, they run to their oncologist. Oh, I want metabolic therapy. Give me. Oh, no, we can't do it. It's not part of the standard care. I never heard of it. We're really good. I would have heard about it in medical school. We have to retrain the entire oncology field to understand what cancer is. That's the problem. You don't have knowledgeable people. What is a guy on the street going to do? Oh my God, I have blood. I have a colon problem. I have a lung. I'm coughing up blood. What is it? He runs down to Dana-Farber, MD Anderson, Sloan Kettering, runs down to these guys, and they nuke and poison the guy. I never heard cancer was a metabolic disease. Well, for Christ's sake, man, get out of the way. You're not moving the field forward. Read the literature. If you can't understand it, I'll be happy to explain it to you if you can't understand it.
Well, can I bring it back to the point that I tried to raise earlier about the patient adoption? Because this is something that I've run into personally as well, which is that even if someone is confronted with the fact that they can make a dietary modification to spare themselves the pain of chemotherapy and probably achieve much better outcomes, there's a certain type of person that can't conceive of not eating bread or candy or soda pop or all of the, these are legitimately addictive substances that actually do get you high, and people are used to it in their daily lives, and it's really, really difficult with a subset of the population to convince them to do that. And so if some person in a white coat is offering them, even a miserable experience like chemotherapy, it somehow seems appealing to a certain portion of the population. And I'm afraid that it's a large portion of the population.
Oh, I agree with you. And if you look at the reasons why those folks from our glioblastoma trial, the ones who did not adhere to the diet, which wasn't a bad diet, it was actually salmon and olive oil and things like this, calorie restricted. They thought that they didn't want to eat the salmon and they take the olive oil. They thought the radiation and chemo was enough. They did enough, and they all died, except one person. So that's the choice of the person. Listen, you can, it's your precious soul. If you don't want to exist on the planet, I've seen it myself. I've had spouses telling the other spouse, stop eating the carbohydrates. This woman died in the hospice with the Hershey bars all over her lap. Yes, you're 100% correct, but those are hopeless people. You can't do it. If they don't care about their soul and their existence, why should we care about it?
I think that population could be winnowed down if the people in white coats were promoting it. I think there's something about the escape hatch of the white coat telling you that that's stupid. On top of it being difficult, if somebody in an authority position isn't reinforcing, I think it really takes coaching is what I'm trying to say, is that if you really want to get somebody in any addictive situation to move beyond that, they're going to need some sort of counseling along the way. You're going to have to be checking in with them. Hey, how's the diet going? I know it's hard, blah, blah, blah. But if the white coat is just saying that's stupid, just take these, toxins.
Well, that's why we developed the glucose ketone index calculator, which is a quantitative measure to allow the patient to know what zone they're in. So, and they could get the data off their cell phone from the Keto Mojo meter. And therefore, they will know whether or not whatever they're doing is helping or hindering. And don't forget, a lot of these chemos that we could use in very low dosages become far more therapeutic. So we don't throw out all the tools of the standard of care. We just reevaluate how they're being used. But the glucose ketone index calculator, which is we developed here at Boston College, allows the patient to know when they have low glucose and elevated ketones to a certain level, a quantitative number, 2.0 and below. As a matter of fact, we're going, and not only that, it works for preventing cancer and chronic disease. because all chronic diseases in one way or another, including cancer, involve mitochondrial dysfunction. So you have now a quantitative measure to reduce risk for chronic diseases, which include a whole range of things, as well as cancer. And if you want to party a little bit, where your number is going into what zone, and how long you want to live in these zones. So your green zone is like 2.0 and below. That'll significantly put pressure on surviving tumor cells based on the glucose part. But you can stay healthy. We call it the maintenance health maintenance zone from about 2 to 12 GKI. Anything above 50, you're putting yourself in an inflammatory zone. So
What is this? How does this calculator work?
you take a blood. It's a glucose ketone meter.
I see.
And right now, you take a prick of your finger and you take a glucose stick and you get a drop of blood and put it into the machine, the meter, the mojo, and it reads what your glucose is in millimolar. And then you squeeze your finger a little bit more and you take the ketone strip and touch it to the drop of blood and you put that into the meter. And then you push the button GKI and it gives you the ratio of blood sugar to blood ketone bodies.
I see. So this is a device that you have, and then you're basically doing like pinprick tests.
Yeah, well, it's just like a diabetic would do,
Yeah.
to maintain. But for cancer and chronic disease management, you can easily put yourself into these new zones, and then you would come to the realization of what you can and cannot eat. If the bread you're eating is highly carbohydrate bread, you're going to see a massive spike in your GKI. But maybe more natural complex carb breads will not do that, and everybody will be will have their own biology, their own numbers, and they'll be always looking on their phone to see what zone they're in based on the glucose ketone index calculator.
Are you passionate about the groundbreaking and heroic research of Dr. Thomas Seyfried on metabolic therapy and cancer? So are we. That's why we've created something special for you, in collaboration with Johnny Rockermeyer, a German book publisher and translator. Introducing our collection of meticulously crafted books that distill the essence of Dr. Seyfried's work. Dive into the science and discoveries. These summary books are your gateway to understanding the intricate world of metabolic ketogenic therapy in a clear, concise, and engaging way. Whether you're new to the subject or a seasoned enthusiast, our books offer insights that can change your life. Ready to explore this transformative knowledge? Visit our website at www.cancerasametabolicdisease.com to get your copy. You can buy the ebook there directly and the paper book via the provided links. Here's the best part. A portion of every purchase goes directly to support Dr. Thomas Seyfried's groundbreaking research. That's why the direct ebook purchase is the best option to donate as much as possible. You can see all of the donations Mr. Rockermeyer has already made at www.ketoforcancer.net. That's right, when you buy our books, you're not just...
But right now, it's only done in cancer patients. But we can prevent. You can't get cancer if your mitochondria remain healthy, and it's hard to get chronic diseases if your mitochondria remain healthy. You have to realize, again, in evolutionary biology, our Paleolithic ancestors were always in a state of nutritional ketosis because there was no high carbohydrate processed foods anywhere to be available. Not if we gave it to them, of course they would eat it, but there wasn't. It wasn't available. You were killing eating animals, mastodons and buffaloes and all this kind of stuff. We were responsible for the extinction of several large species because we killed and ate them. So and there's not a lot of carbohydrate in the beef liver or in the marrow of a mastodon. So our ancestors were always in a state of nutritional ketosis, not because they wanted to be, because they didn't really have any choices. It was our technology that continued to improve during the Neolithic period, developing corn, wheat, rice. Civilizations depended on the fact that we didn't have to hunt and gather any longer. But that put us then at risk for different kinds of chronic diseases, which are exploding today because we have internationally highly processed foods. I got a Twinkie here that I always do in my class. This Twinkie, if you see, they look pretty good, right? You see them there?
Mm.
Okay, these are 12-year-old Twinkies. All right, they're a little stale. A mouse broke in last year and ate one, So he didn't seem to be concerned about its age. But the issue is we're eating this crap, and then we wonder why we have diabetes, cancer, hypertension, cardiovascular disease, all this stuff. And because our ancestors never had this, not that they wouldn't want it, but they died from infections and injuries. We didn't have orthopedic surgeons, and we didn't have antibiotics. So they died from injury and infections. Where today,
Are you ever tempted to eat one of those Twinkies late at night in the office?
no,
Everything's closed?
not the 12-year-old one. But if you go to Atlanta, there's a bar in central Atlanta, Central City, called the Nook, and they take fresh Twinkies and deep fry them, and
Wow.
put chocolate syrup and powdered sugar on top of it. And people are knocking each other down to get to these Twinkies.
That's unbelievable. I mean, I'm sure
Irresistible.
it's tasty. I think
Oh,
that
yeah.
they
Yeah.
also do like deep fried Mars bars at
Yes,
state
all
fairs
that.
and stuff like that.
Well, the
You
thing
have
of
a
it
whole
is,
box.
I bought it. When Hostess ran out, they were going to sell 10 years ago. I said I better get some just to have it as a prop for my class.
But seriously, there's kind of a dark thread to this, right? Where you essentially have the advent of agricultural populations, which sustains essentially a slave class. So you can have these huge armies and so forth. It's very cheap compared to herding animals around, and you can, and so we're in a situation that still persists where eating a ketogenic diet can be a little on the expensive side of things. Like I myself have been doing it for about a year now, for not because I had cancer or anything, but for some other reasons that I wanted to try to improve in my own health. And it does tend to cost a little bit more from like eating meat, like especially good stuff, right? If you want to get like really, if you really want to try to find, locally raised stuff and like, healthy sources of all of this, and also ecologically friendly sources, it's not cheap. And I wonder if, you can buy a pizza for what, $10 or something, and get your whole caloric content for the day very quickly. And so I worry that there's a side of this as well that pushes people away from it, just because it's in some sense economically unplausible for a lot of people.
Oh, you're 100% correct. Because in some underserved populations, they have food deserts, and the only food available is highly processed carbohydrates, putting them at risk for all these chronic diseases. And if they want to go outside the food desert to get a nutritious diet with low carbohydrate, it costs them. They can't afford it, so they eat the high processed foods, whether it's donuts, hamburgers, pizzas, or whatever. And when you do metabolic therapy, if you have acute cancer, you might want to consider not eating some of that until you put your tumor in check. But the goal is for us never to say what you can and cannot eat. No government or people should say that. It makes people want to eat it more, or whatever. But just to be aware, and a lot of people aren't even aware of this. The federal government is not issuing any warning signs at all. I know Michelle Obama did. She tried to do that, to get out and exercise a little bit more, eat a little bit more fresh stuff, and then she was kind of vilified by the industry for doing that, which is again immoral. She was saying the right thing, and they kind of discounted that. But with a glucose ketone index, you can now know better, and that doesn't cost. The meter is cheap. And now we're developing free Libre. You can have these arm patches that will give you real-time blood sugar. And as the technology improves, you'll have real-time ketones, and you'll be able to do the GKI directly on your arm, so know where you are at all times. And then
I got these little just urine sticks for the
yeah.
ketones that I don't know how effective or accurate they are, but they do seem to, in some sense, at least grossly tell you whether you're in ketosis or not.
Yeah, that's true. They do. And we always say, because the ketone strips are more expensive than the glucose strips for the meter. So you can use your urine strips for the in-between times. But we're at this technology of monitoring physiological health on cell phones from arm patches and things like this, at least give people an idea to choose how they want to live and what they want to do. And if that could be integrated with coaching, and folks and the medical establishment all working together, you're going to drop chronic diseases and you're going to drop death from cancer for sure, because we can use metabolic therapy not only to prevent cancer, but once you have cancer, then we just modify the treatments in a little bit different way to kill tumor cells rather than keeping normal cells healthy. So you keep normal cells healthy, you prevent cancer. But if you have cancer, you can still use the same ketone body to replace glucose, and then target the tumor cells. So it's really quite flexible, and it's all going to come down the pike, because we can't continue to live the way we're doing. And we have to modify it for the very questions that and issues that you guys are raising. But without any knowledge at all and having no clue about what then everybody's lost. They don't know what to do. Can I eat this? Can I eat that? What about this? What about that? And then you run into all these charlatans who say, oh, take this supplement, that supplement. These guys have no clue what they're doing. You have to have some scientific credibility to know what's going on. So all of this is the future. What I'm telling you right now is the future, because we're not doing it today.
Pardon the interruption, but I have to ask you for a favor. If you are listening to this podcast and you like what you hear and you would like to support the project, please come over to patreon.com/DemystifySci. For just 75 cents a week, which is not very much at all, you can help us keep the ship going indefinitely. You can help us go record conversations in person. You can help us plan future conferences. You can help us turn the paradigm around and bring forth a scientific revolution, the likes of which the world has never seen. I'll see you over at patreon.com. Let's talk about this fembendazole, mebendazole potential adjuvant to ketogenic diet, because while you guys were talking, I looked up a paper from anti-cancer research. It looks like it might be a Vietnamese group that basically published on oral fembendazole for cancer therapy in humans and animals. And early on in the paper, they mentioned that fembendazole garnered global attention as a potential anti-cancer therapy following the complete recovery success story of Joe Tippins, who was diagnosed with small cell lung cancer. He was in a clinical trial for a novel anti-cancer drug, but under the guidance of a veterinarian, started self-administering 200 milligrams of oral fembendazole, vitamin E supplements, CBD oil, and curcumin, and after 3 months of self-administration, a PET scan revealed no detectable cancer cells in his body. And he was the only patient cured among the 1,000 patients in the study. Everybody's like, anecdata, plural of anecdote, is not data. And if you start looking at, fembendazole, mebendazole mechanisms of action, they're tubulin inhibitors, which is this like structural component of parasite cells. So can we take apart the biomechanics of these compounds inside of cancer cells? Like, what are they doing?
Yeah, well, the tubulin hypothesis was put out, but you have to also realize that the formation of tubulins requires a lot of energy. So ATP, again, we're back to energy. Without energy, nothing can live, nothing can form, nothing can do anything without energy. We have a paper that was a bioarchives paper, and we're working on it here. We've shown that mbendazole, which is very similar to fenbendazole, targets the ability of the cell to use sugar, glucose, and also disrupts glutaminolysis pathway, which is the glutamine. It's not as powerful as DON, but it's doing the same thing. That's what's going on with those things. The tubulin issue, because you have vincristin, which is a very toxic chemo, and that targets tubulin, and you get sick as a dog. But if fenbendazole is working because it's targeting tubulin, how come you're not so sick like you would be if you took vincristin? What is fenbendazole doing? So in India, where they did a lot of this work because of their parasites, the large number of members of their population have parasites. So there was a lot of basic research over decades on these kinds of anti-parasitic drugs. And between the Chinese and the Indians, they showed that fenbendazole and mbendazole block glucose transporters. Wow. So you're going to inhibit a little bit on so that's going to block down glycolysis, which is cytosolic substrate level phosphorylation, one of the two fermentation pathways driving the dysregulated growth. And our new findings that we're working on next door as I speak is showing that this mbendazole also disrupted the glutaminolysis pathway, which is the fermentation of glutamine. So we're seeing exactly what we've been saying. So why those things work is because, and then you put the patient into nutritional ketosis while you're giving these parasite medications. It works even better without toxicity. So yeah, we're working it. We published that, but it's not. It's bioarchives. It was we developed a therapy for pediatric brain cancer, which if they used it, they could. Well, we'd have. It's tragic what we're doing to these little kids in the brain with brain tumors, irradiating, hacking them apart, and everything. When all we have to do is put them on fenbendazole or mbendazole in calorie-restricted ketogenic diets, and manage it that way. But boy, they would never touch that with it. You know what's really weird is that if you have a child with epilepsy, they're on one part of the corridor. You treat children with epilepsy with ketogenic metabolic therapy. But God forbid, just down the corridor, they're irradiating and poisoning these little kids with brain tumors when they could be using mbendazole, fenbendazole. They could be using ketogenic metabolic therapy for managing the cancer in these little kids as well. This is the craziness of the medical profession. It's like, what the hell? And it took years before it took the work of Jim Abrams, the movie maker, with Meryl Streep and others, to bring attention to the fact that you can manage epilepsy with diet and lifestyle issues. But no, you can't do that for cancer. This is taboo going down that direction. So, but people want to live and they don't want to be brutalized. And you got to have a knowledgeable group of people. So when you're talking about Joe Tippins, yeah, Joe used a fenbendazole, and cocktails of parasite medications work real well. But you can't make any money on that. This is old El Cheapo stuff. And don't forget, say it, revenue generation first, patient outcome second. Say it, revenue generation first, patient outcome second. So we have
remember that there was a one of the consulting firms or some investment firm put out a brief that was talking about business models for the medical industry. And the title of the presentation was, "Is Curing Patients a Sustainable Business Strategy?" And I'm like, no, probably not. I mean, if you can just basically give people something that they can do at home and some kind of cheapo medicine.
Yeah, well, I think we have to, we're not against revenue generation. I just don't know how to do that. My job is to kill cancer cells without toxicity. That's what I know how to do.
But it might be that it's
I'm leaving
not
it to
a
somebody
billion-dollar
else.
business.
Yeah, well, but somebody's going to figure it out. We're a very innovative species. If there's money to be made, we'll figure out how to make it. And but I'll leave it to those entrepreneur type people. I don't. Our goal is just to see how many. Because if you're right, then the overall outcome should be survival with high quality of life, or program. What do they call it? Progression-free survival and overall survival should be significantly improved. I get a lot of enjoyment out of that. If I see some guy who did metabolic therapy and his physician told him, "Oh, you have 9 months to live," and he's at 5 years later because he did metabolic therapy according to our program, I feel good about that. I don't need to make $1 billion on something like that. But it lets us know we're on the right path. So I'll let somebody else figure out how you're going to make $1 billion on that. But that's their goal, because that's what we are. We're very innovative in what we can do, and how to make a buck there's always that kind of a guy. So but you always want it to be available for the underserved people. They're getting hammered unnecessarily. The poor and underserved folks, the minority groups, and those guys, they're really being brutalized. So and because they don't know, number one, there's an educational issue, there's an exploitation issue, there's a lot of other kinds of issues, availability of things issues. So yeah, so that lets me know that if we're on the right path and we can keep people alive, it shouldn't be oh, this guy lived with a GBM for 4.5 years. Oh my God. No, that should be the standard, not the exception. So we got Pablo
I did discover
Kelly.
a really funny loophole. I had to present at APS, the physics conference last year. And so I had to eat out for several days. I was in Minnesota. And I discovered very quickly that there's a strange loophole at restaurants where if you go to like a hamburger place or something, right? You order the hamburger, and of course, if you're eating keto, you don't get the bun, right? But the funny thing is, they don't really have anything in their computers for the most part. If you want to add an extra hamburger patty, it's basically $1 or $2. And so you can eat a lot of meat at restaurants by ordering an extra chicken patty, an extra hamburger patty. It's really quite hilarious. I'm sure it's terrible quality meat, but I mean, it is a funny loophole.
Next time you go to Minnesota, go to Manny's. Manny's Steakhouse. Have you been there yet?
I
Oh,
don't think that I went there.
oh
No,
well.
it's good
I'm
stuff.
not. I mean, that's the biggest steaks you're going to find. Tomahawk ribeyes and these big steaks, unbelievable.
I did go to this
So,
Brazilian steakhouse that was pretty amazing.
yeah,
Um, yeah, I've never
yeah.
been to anything
Well,
like that before.
well, a steak, high quality beef, again, is good for keeping low GKI, low glucose ketone index. Chicken, and you can like the people in that breast cancer, brain cancer study that you speak of. If you saw their diet, it was salmon, olive oil, a little avocado, a little bit of vegetable, but a lot of that was pretty much what it was. It wasn't so dramatically different that people couldn't eat it. You could easily substitute the salmon with a ribeye, So it all is to keep your GKI. What's going to move your GKI down in the zone, right? And if it's a big ribeye, yeah, oh wow, I can eat a ribeye. Nobody, oh, well, I'm a vegan. Well, I don't know about the vegans. They have to work a little harder to get into the GKI. They have to take a lot of supplements and other things where a carnivore guy could probably get more. It's easier to do, but I don't push carnivore, vegan. I don't care what pizza pie. You get a GKI that's low enough, you'll be in a better zone. And whatever that person needs to do to get into that zone, my hat's off to them.
I like
So
that.
so we don't profess any kind of a particular diet. All we know is that eating plants alone is more difficult to lower your GKI.
Hmm.
Sure.
And if people don't believe me, then they go out and see what their GK. If some guy wants to show me a GKI of 0.6, eating broccoli all day long, then I'll say, wow, I'm educated now. But so far we haven't seen that.
How much variability are we discovering among people's metabolic processing? So, because conventional wisdom says that if you want to have this relatively good glucose to ketone ratio, you're probably going to have to eat animal products because they're the ones that have the fewest carbohydrates. Are there people that just have radically different metabolic strategies where they're able to keep their blood glucose down eating things that are starchier? Or are we finding as we collect more data that this is actually a decent model for like all humans?
Yeah, well, I don't know because all this is quite new,
Mm.
and we don't have epidemiological studies on glucose GKI values like vegans and guys that eat only fish, guys that eat only beef. What does it do? I think that's all the future because right now we don't have the availability of the technology. That we have the Precision Extra meter from Abbott, or you have the Keto Mojo meter. We're developing the free Libras. Then everybody will be able to collect the data on themselves and see if we do in fact have groups of folks that fall into different categories with respect to what they eat and what effect it has on their GKI. And people will naturally want to experiment on themselves to see the ranges of what they are or unable or are not able to eat, and that's all going to be very exciting. But we are a obsessive compulsive species as well. Everybody wants to go out and better the other guy who can get the lowest GKI. It's true. And they're going to so we have to be cautious that we don't overdo it in a lot of ways. But I think it's going to be an exciting opportunity for the future to integrate what you guys are saying, but also linking it to prevention and management of cancer as well as chronic diseases, because now we have tools that we can do it. So it's not so mysterious. Eat this and you're going to get that. Eat that. Don't eat this. Don't eat that. What the hell? Just do your GKI. You'll know what you can and cannot.
Yeah.
So,
So, are there any downsides to this kind of diet in general?
Exactly what I was going to ask.
well, I mean, I would say that we as a species evolved to be in ketosis. So that's who we are for hundreds of thousands of years. We were in a ketotic state. We had a lot of exercise. We didn't have any highly processed carbohydrates in our diet. You can only get, it's hard to get obese eating buffalo meat all day long. It's eating elk, eating elephant, okay, woolly mammoths. How many how could fat could you get by eating woolly mammoth bone marrow all day long? And we also have a hormone in our body called cholecystokinin. It's an appetite suppressant hormone that affects the 10th cranial nerve, and it controls appetite. So when you eat a lot of protein and fat, you upregulate cholecystokinin, and you don't feel hungry anymore. So interestingly enough, when you eat high carbohydrates, you kind of reduce cholecystokinin activity, and you feel hungry all the time. So yo-yo dieting and all this kind of crazy stuff. And then again, you definitely lose weight with exercise and eating in ketosis. There's no question about it. This is because we didn't see obese. Paleolithic man was definitely not obese. And then when we look at modern people today, those folks that live according to traditional ways, almost invariably, they have very little cancer. They're lean, very fit looking kinds of people. They may have parasites and other kinds of conditions, but they're not obese, and generally they don't suffer from chronic diseases. So you have to put all that in perspective. But it's going to be exciting to see once this becomes more available and streamlined, how people are going to have these GKI contests. And then there's the other group who can get the highest GKI.
Uh-oh.
Okay, so right, not that anybody would want that. Who the hell would want to eat like 100 hot dogs in 10 minutes? I mean, people, but yet Joey Chestnut's out there doing this kind of stuff. So people can say, "Well, let me see if I can eat 12 pizzas and wash it down with a gallon of sweet tea to see if I can jack up my GKI." I think the highest one we've seen was in a diabetic person, maybe 1,000 something at GKI, but it's usually associated with some metabolic imbalance of some sort. So, but you got nuts like this. I mean, let's be honest, we're not a logical species in any way.
No, we are not. I wanted to ask you about the exercise component of this. So we have the ketosis, we have metabolic therapy by the application of something that interrupts glutamine metabolism. How much exercise do you need? Are these people running like a marathon a day? Is it enough to take a walk? Like, what are we talking about?
Yeah, well, we know that glutamine builds muscles. So if you exercise, the glutamine in the blood, to some extent, will be reduced. Um, we also know that when muscles are contracted in aerobic exercise, they take the glucose out of the bloodstream. Muscle does not share its glucose with any other organ. This is another aspect of evolutionary biology. Liver will share because it turns the glucose. It can produce gluconeogenesis from other sources. Once the stored glycogen is used up, it will take some amino acids and things to produce glucose at a very low level. Because erythrocytes, you got to have the erythrocytes. They always need some small level of glucose. But exercise, okay. So why evolutionary biology? Because we have to think back. We were under threat from being killed by another member of our species or eaten by some animal. If you didn't have any energy to run away or defend yourself, you would have been victimized. So we need the energy, and our muscles, we have to run and fight, and the muscles have the energy to do that. So the muscle isn't going to share, oh, my brain needs some sugar. Let's give all the glycogen from my muscles to the brain. And when the tiger comes, you're his meal. That's evolutionary biology. But but exercising will pull glucose out of the bloodstream. And if you're already in a ketotic state, you can enhance this a little bit more. Also, appetite. You're not thinking about food when you're walking. Now, I'm not saying what kind of exercise. It could be aerobic walking. If you're young, you could be running. You could be lifting weights. There's a bunch swimming, biking. Doesn't make any difference. As long as the muscles are active, they're going to be pulling glucose out of the bloodstream. So if you have cancer and you're on a low carbohydrate diet and you're exercising and you're taking fenbendazole or embendazole, man, you're going to put a world of hurt on these damn tumor cells. And then you elevate your ketones, which the normal cells in the brain can use, and the muscles and the heart, but not to the tumor cell. Tumor cells can't use that because they have a poor mitochondria. You need a good mitochondria to manage fatty acids and ketone bodies. So, and we interrogated all. We did all these experiments in the lab, so we know pretty much what's going to happen.
Do you have a feeling that a lot of the lack of response is due to there being a long history of charlatan propositions for how to cure cancer. Like famously, Steve Jobs died of pancreatic cancer because instead of getting chemotherapy, he followed the advice of somebody who I think put him on like a fruit juice diet.
Yeah, well, see, we now know he didn't know at that time, nor did I. I didn't know what was, I mean, other than the glucose, we weren't really clear about the glutamine. Warburg himself didn't know about that. And I have no idea what that diet that Steve Jobs took did to his glucose ketone index. He never measured. We didn't have it then. So we can only speculate about what happened. I don't know.
Well, and I'm not specifically asking about Jobs. I'm asking more about the sense that I feel like people are really attached to this idea that the standard of care of radiation, of chemotherapy, of these really aggressive treatments is the only thing that can help. And there's a vibe when you talk to people where you're like, hey, have you considered that there's other possibilities? Like Shiloh says, like we talk to people who we have friends that have cancer and we're like, I have a friend who's an ultra marathon runner. And this is a grown ass man who keeps a bag of gummy bears in his car
Yeah.
because that's just what he's used to from ultra endurance running, where everybody's just eating sugar snacks all day long. And he's basically like, well, the standard of care is chemo and radiation.
Yeah, well, we got 1,700 people a day dying, and goes up every year, and more diagnosis. I'm seeing a lot of young people now coming to me, young, late 20s and mid 30s, early 40s. So I said, what the hell is going on? So clearly we have an obesity epidemic. Let's be honest. If people knew that obesity is replacing smoking as a major risk factor, but they don't know that. So there's a massive lack of knowledge, I think, on the part of the scientists who think cancer is a genetic disease, and they're constantly hunting for gene mutations that might be responsible for your dysregulated cell growth. There's a pharmaceutical industry making billions and billions of dollars coming out with immunotherapies or some other kind of therapy dependent in large part on the somatic mutation theory, which has now been undermined, discredited massively. And then you have the hospital industry, which generates tremendous revenue for radiation and chemo and this other stuff. And then you have the federal government that says cancer is a genetic disease, awarding research grants to investigators at top medical schools and science centers looking for mutations in genes that might be responsible for your dysregulated cell growth. So you have mis and disinformation at every level in the cancer industry.
But can we be clear though, like there are genetic changes associated with cancer. You're just saying that they're downstream of the metabolic issues. Is that correct?
There's two issues. There's one, somatic mutations, which is called the somatic mutation theory. This is where mutations of lifestyle or living cause nuclear gene mutations. Then there's germline mutations. And you hear about the BRCA one with Angelina Jolie. She's made that kind of famous, having her breasts and ovaries removed or something like this. And then you have the Li-Fraumeni, the Lynch syndrome. These are germline mutations. And we have a paper just ready to submit with my colleague, Bob Kaplan, where we went through every major syndrome, inherited germline syndrome, and every one of those inherited mutations disrupts oxidative phosphorylation in the mitochondria. So, and none of them are 100% penetrant, meaning that a person could have the mutation, like BRCA1, it's like 60% penetrant, meaning 40% of the women that would have that mutation never develop cancer. They only develop cancer if the mutation disrupts oxidative phosphorylation, leading to substrate level phosphorylation and dysregulated cell growth. So there's secondary risk factors. The primary risk factor for all cancer, all major cancers, is insufficient oxidative phosphorylation coupled to a compensatory substrate level phosphorylation, leading to dysregulated cell growth. Not that complicated. So, but for a lot of people, it's overwhelmingly complicated. It's less complicated than people made it out to be. So you just have to know what the inherited risk factor is no different than when we're exposed to a chemical carcinogen or oncogenic virus, hepatitis C, papillomaviruses. I showed how those viruses are linked to dysregulated cell growth through an effect on oxidative phosphorylation. So the virus or the product of the virus goes to the mitochondria, disrupts sufficient oxidative phosphorylation, leading to chronic upregulation of substrate level phosphorylation and dysregulated cell growth, driven by fermentation, glucose, and glutamine. Every major cancer is doing the same damn thing. So it's one disease. It's not 100 different diseases. It's the common pathophysiological mechanism is insufficient oxidative phosphorylation coupled to increased substrate level phosphorylation, facilitated by oncogenes, HIF1 alpha, MYC, open the floodgates to get glucose and glutamine into the cell. So those oncogenes are just facilitators of driving the substrate level phosphorylation. We have that paper that's going to come out in our big paper. So all of this is there. We publish it open access. Everybody is welcome to read it. If they choose to ignore it. So you can't do anything about that.
Have you heard of Stephanie Seneff's work?
I've heard of her. Yes, I've heard some part. I'm not intimately familiar with that. I think she's down at MIT or something.
Yeah, and she's been doing a lot of really interesting work on the effect of glyphosates on disrupting metabolic function, where her mechanism is that glyphosate interrupts the dehydrogenases that are important for processing the intermediates for the mitochondria. And so when the enzymes that are packaging basically the carbon compounds that the mitochondria needs to function are broken down, you get all of these downstream effects.
Yeah, well, I think that could possibly be certainly linked to the oncogenic paradox. So it would fall into that line. Again, the origin can be one person could get breast cancer from a different reason from another person, but the bottom line is all the cells are fermenting, and regardless of what particular mechanism was responsible for the chronic disruption of oxidative phosphorylation, you're dealing with a population of cells that are fermenting. So now you have to deal with that, which is then goes into the management section. So we can try to avoid cancer by avoiding the provocative agents that would lead to disruption of OXPHOS. But once you have cancer, now you have to deal with the fuels that are driving it.
Mm.
Yeah, I was going to ask just about the different mechanisms of injury to the oxidative phosphorylation system. Like, what are the different ways that can get disturbed?
Well, when you look at carcinogens, they call these molecules carcinogens, and because they cause cancer. And the question is, oh yeah, I see a lot of DNA mutations when I look at a tumor that was caused by a carcinogen. But the origin of how that dysregulated cell growth came in the first place, if you look at, there was a beautiful study, I have it in my book, where they looked at these carcinogen exposed cells, and they're bioluminescent, they're fluorescent. The inside the mitochondria are like glittering, which is from the reactive oxygen species, and that's where the mutations largely come from. When oxidative phosphorylation is not efficient, ROS, reactive oxygen species, are the product of dysregulated oxidative phosphorylation. And these ROS are carcinogenic and mutagenic. They further damage the lipids and the proteins of the mitochondria, and they cause nuclear genetic instability and other kinds of mutations, as you see as downstream effects.
And repair is really costly as well, right? The enzymes that have to come in and fix these things need a solid source of ATP.
Yeah, and the cancer cell seems to be buffered from the ROS, because a major reducer of reactive oxygen species is the glutathione system, and the glutathione system is part of the the pentose phosphate pathway, producing a large amount of NADPH that drives this what we call antioxidant system. And the carbons from glucose and the amino acid glutamate are part of the glutathione system. So it's unbelievable. So the two fuels that are driving the dysregulated growth also protect the tumor cell from its own damage, from the damage of the ROS, but the nucleus of the cell is being damaged. So you're collecting DNA mutations as a downstream effect of oxidative phosphorylation, and people are all focusing on the downstream effects rather than the original, the fuel that's driving the beast. So once we know that, we know how to put out the fire. We know how to slowly degrade or interrupt the two fuels driving the dysregulated growth and keeping the rest of the body healthy. So when normal cells are burning ketone bodies, we produce much fewer ROS. And the bioenergetic mechanism of that was worked out with my late friend Richard Veach from the NIH, where ketone bodies do not produce FADH2. They produce only NADH, delivering electrons to complex 1 and keeping the coenzyme Q coupled in a fully oxidized state. Because that's where the ROS are coming from the half reduced form. And when you start throwing more and more ROS out, you have a tendency to degrade the efficiency of your energetic system. So burning ketone bodies is a way to protect the integrity of your mitochondria, maintaining a highly efficient energy state, keeping you from not only cancer, but numerous other chronic diseases. And also life. You live longer when you keep the ROS restricted. So it's aging, anti-aging, anti-cancer, anti-chronic disease, but nobody wants to do it.
I mean, it's a
They'd rather,
hell of a.
they'd rather eat that, right? This is as long as you know that. See, when you're completely unknowledgeable, then you would never know what I'm telling you. But now you actually know and have a mechanism that can explain it. Now it's your choice to say whether I want to engage or not engage in these activities.
Do you think that there's cancers that this doesn't apply to?
Haven't found one yet. So, and our papers show all major. So when I publish these cancers data on the damage to mitochondria, lipid drop accumulation, these are all biomarkers of dysregulated cell growth. These are all biomarkers of inefficient oxidative phosphorylation. 98% of all cancers fall into this. Is there a cancer out there that can grow out of control with normal oxidative phosphorylation and not dependent on substrate level phosphorylation? I've offered $100 to my students for the last 5 years, a crisp $100 bill. I'll give it to you. You find a tumor that can grow in a dysregulated way without a dependency on substrate level phosphorylation. I'll give you the $100, no problem.
Throw a Twinkie in and they might be able to do it.
No, they ain't going to do it with a Twinkie. Because the Twinkie's loaded with sugar, you got to do it with a cancer cell that's respiring without fermentation and growing out of it. Now, interesting thing is liver regeneration can grow as fast. Regenerating liver cells can grow just as fast as tumor cells. So you have a hepatoma, which is a liver cancer. Okay, that will permeate your whole liver and kill you. Well, if you take a lobe off your liver, the liver naturally regenerates very rapidly. And they are using glucose and glutamine in very low quantities, but they're respiring those two fuels. They're not fermenting those two fuels. So it's very interesting to see that. And the liver knows when to stop. Liver regeneration knows it just doesn't keep developing more and more new lobes of liver for yourself. They know when to stop. But the hepatoma does not know when to stop and ends up killing you.
Hey, who else is working on this? Are you completely alone? You mentioned your colleague, Robert. I couldn't remember his last name.
Yeah, well, Bob Kaplan, he's just an associate with us. He's doing he's working very hard on this. But Christos Xenopoulos in Semmelweis University, the world leader on mitochondrial substrate level phosphorylation. When you don't know certain things, you reach out to those individuals that are the experts in those areas. You then introduce them to the problem. They start looking at it with their level of expertise, and then you bring the groups together to do this. We have a lot of physicians that are joining who are reading about what we're doing because they're into the issue of patient care. So they want to use ketogenic metabolic therapy to look at the outcome of their patients. But these are in small clinics and isolated conditions. They're not at the top medical schools, the top cancer research centers. They're not. These guys are all locked into the somatic mutation theory, lockstep with whatever the NIH says. So whatever the NIH says, whatever the standard of care says, they're in lockstep with that. And they're not willing or capable or even desirable to change their way of doing things.
How do you get grant funding if you can't rely on those traditional structures?
All of our funding comes from philanthropy and private foundations. So
Wow.
Folks make the donations to the University, Boston College, saying, I want to support Seyfried's research. So we get donations directly because I cannot accept a nickel from anybody. It has to come through legitimate channels. The university has a channel here. And then we have private foundations, Travis Christofferson's Foundation for Cancer Metabolic Therapies, and then private foundations are reaching out slowly to us, because why should you dump in thousands and thousands, potentially millions of dollars into these programs that haven't done anything. There's no accountability. Where the hell does all the money go? Susan Komen, breast cancer found people running and swimming and jumping and climbing, and all we do is get more dead people. There doesn't seem. And then when you look at what they're doing, they're hunting for genes, and you say, now I know why you get all these dead people. The only people getting healthy are the ones running and jumping and whatever the foundation is, But the poor people are getting brutalized. So 1,700 people a day. And you can raise money for the lung cancer foundation, the breast cancer, the brain cancer, and then you do standard of care and get brutalized. So and they feel good raising money for a failed operation. So
now that you mentioned standard of care, I remembered I was going to ask you: is it possible to run a trial of your protocol without also having to give somebody radiation or chemo?
Well, that's what our hope is. But we were slapped down at Cedars-Sinai. They wouldn't do it because Jethro Yu is working with us, and he said they'll do metabolic ketogenic only after you do standard of care. Well, standard of care is only put you on the slope to death. So now you're going to try to take and not only keep a guy alive, but try to correct all the damage that radiation and poisoning did to the body. This makes no sense. Why don't you start with metabolic therapy, and then if that doesn't do a real good job, you might want to consider a little temozolomide or something like that, rather than poisoning and irradiating the guy up front. Why don't you consider that as a secondary option after you can? Oh, the tumor shrunk. I don't know what happened. Yeah, now you want to irradiate this poor guy when the tumor is shrinking, and the surgeon can say, "I think I can get it all out." I mean, everything is back ass forward. And it takes knowledge. People have to become familiar with what I'm saying, and we're publishing this. It's not like I'm telling you this. Go and read the papers over and over again. Open access papers. We got them out there. People can read.
You said you have a book out also?
Yeah, well, my book is Cancer as a Metabolic Disease. I wrote that book in 2012. Cancer as a Metabolic Disease. I speculated on some things. I had an idea about mitochondrial substrate level phosphorylation as being the second arm of the fermentation pathway. But then I got Christos Xenopoulos to work with me to prove it. And that paper came out in ASN Neuro for brain glioblastoma. But now we're starting to see it for all these different cancers. So it's just a matter of time. I don't know how long it's going to take, but we're not going anywhere. As long as philanthropy and private foundations support us, we're going to pound down this research. They choke on it. So you have no choice but to accept it.
Do you think that, do you aim for publishing in a place like Nature or Science or Cell, or are you more preoccupied with just getting the work out there?
Well, I publish in peer-reviewed good journals. Cell and Science today have become overly involved with minutia. They love to have supplement after supplement after supplement. Oh, you didn't do this. You didn't do that. You
figures
didn't.
1 through 14.
People living twice as long, man. Huh?
Yeah, I said figures 1 through 14 with panels A through J for each figure.
The little tiny Western blots and gene expression, beehive plots, waterfall plots, all these kinds of volcano plots. And, that must be really important, deep. We got 1,700 people a day dying. You're wasting your time on this kind of bullshit. Let me show. And then we got the Greek study. Oh, I can't believe that we had this tremendous success. There must be something wrong with that study.
Yeah, well, I guess the reason that I ask is because I'm like, I know that people in the field respond to things that are published in these big journals, right? Like, we know physicians, we know surgeons, and they're like, oh, well, it wasn't in Nature?
Yeah,
Is it
right.
is it really that reputable? And so, like, I wonder, do you use that as a strategy where you have some, art of war level plan for getting in?
Yeah, no, that's a very good point. But my friend Greg Glassman, the former owner of CrossFit, he runs the Broken Science Initiative. We found that half the stuff in Science and Nature can't be reproduced. This is one of the great problems. Nobody can reproduce that crap. Nobody has enough time to look at. First thing you say is the study in Science and Nature in cancer based on the somatic mutation theory or the mitochondrial metabolic theory. Let's look at it that way right off the bat. If it's in the somatic mutation theory, get rid of it. It's not going to have any relevance to the clinic anyway. So then you start looking at the overall outcome in preclinical systems. How long did you keep your subjects alive? were they syngeneic orthotopic models, meaning the tumor genetics and the host genetics are the same, not in these genetically created monstrosities that we're doing. So you have to look at the systems under which you're working.
You're talking about xenograft tumors where they grow the cancer cells somewhere else and then they inject the animals with them in order to demonstrate that the tumor that they've created can be treated with the drugs that they're using?
Yeah, well, a xenograft, you're taking a human cell and you're growing the human cell in a mouse that has a broken immune system, that has type 2 and type 1 diabetes. You got 50 million years of evolutionary difference between the biology of the human cell and the host mouse. And then you get some revelation. Whoa, look what happened. And you put it in the clinic and it doesn't work. Those should be the last models you look at. The models you should look at are all natural ones, where the tumors arose in that strain of mouse, and it's a natural arising. See if you can manage those kinds of tumors, not some of these genetically engineered things. Yes, they're great technological improvements. For what, I don't know, but it's certainly not dropping the death rate. We got 1,700. If you knew what you were doing and you had a model that was really powerful, we wouldn't have 1,700 people a day dying. So that tells you something's wrong with whatever system, whatever program, whatever you're doing ain't working. And we know that metabolic therapy can drop that death rate very, very significantly. And we have the understanding of the mechanisms responsible for that. But it's too radical right now. Too radical. So we got to wait for the field to catch up with what you know.
I mean, I have a lot of hope. There's somebody else in Boston at Tufts, Michael Levin. Are you familiar with his
Yes,
work?
I know Michael. Yeah, very good.
I think that the ideas that he's putting forth, which is that perhaps there's a higher abstraction level of biology that is not at the level of pure nucleotides inside the genome being dysfunctional, but there being a different approach. If he's successful at what he's trying to do, which is basically regenerative medicine, I think that might be the push that's necessary to get people to start thinking about these questions at the level that you're thinking about them, which is one abstraction layer above, like, what's happening to the genes. It's like, well, there's this functional feature of the cell that's broken, and that's causing everything downstream. Because, like you said earlier, you're like, there's a million different pathways to cancer if you're looking at gene mutations. And it seems like it's a bunch of different disorders. Like, I've heard this in the introductory paragraph of cancer papers, right? Yeah. Cancer is not a uniform disease. It's a bunch of different things. The etiology for each condition is different. And in the back of my mind, I've always been like, that's really weird. That seems like it would be downstream of like a single feature.
Yeah, you're right. So, but every one of those things that has a terribly different genetic or completely different, very different genetic background, they're all fermenters. So that's the commonality of all things. The common pathophysiological mechanism is what, as I said, the replacement of OXPHOS with substrate level phosphorylation, regardless of what the genetic profile tells us. Yeah, Mike Levin and I had discussions. As a matter of fact, I cited his work in my 2015 paper on metastasis, because metastatic cancer arises from fusion hybridization of our immune cell macrophage with a cancer stem cell. And that has the capacity to spread around your body, and you don't need new mutations for that. I told Michael that. He knows that. So we know what metastatic, and those metastatic cancer cells are glutamine driven. So they need the glutamine. So we know how to manage all the metastatic cancers. I mean,
I just wait? That's really, really profound. I want to make sure that everybody heard that. So you're saying that essentially transformed immune cells become the mode by which cancer spreads throughout the body.
Yes, publish that. And listen, here's another thing. I published that called The Origin of Metastatic Cancer. That's my most highly cited paper, 1,230 citations that detail the origin of metastatic cancer as part of the immune system, right? Wow, you think that somebody would catch on to it? In the beginning of the paper, I said cancer metastasis is largely responsible for cancer deaths. That's all they read. So they cite the paper because I made that stupid little sentence, and they never read the damn paper. And then they go and tell us how the epithelial mesenchymal transition, which is based on the somatic mutation, which is I call the werewolf hypothesis. It's just
sorry.
I have it in my book.
You're
It
too
makes no
funny
sense.
sometimes, man.
It makes no damn sense, evolutionarily or any other way you want to look at it. Yet, when you go to the ACR, American Association of Cancer Research meetings, row after row of people with the EMT, MET, I said, don't these people have any functional brain cells? This doesn't make any sense evolutionarily.
So how does that change the approach to metastasis?
Well, now if it's glutamine and glucose dependent, you can kill metastatic cancer. So we came to this revelation because I had a model of brain cancer from Edinburgh. It was called the VM. They form natural tumors in the brain. And then I said, it took me my students and I, over 10 years to figure out what the metastatic cell was. And it turned out to be a macrophage. And then we did a deep dive on all that. I wrote a paper in that review, Origin of Metastatic Cancer, where my student, Leanne Heisentreit, we went through all metastatic cancers and saw that they manufacture markers of macrophages, indicating that there was some sort of fusion hybrid condition. And Akil, in 1905, a German pathologist, said he thinks metastasis comes from some sort of fusion hybrid. The late John Pawlik at Yale was jumping up and down, screaming and yelling about cancer being some sort of fusion hybrid condition. Melissa Wong from Oregon Health Science Center has been saying this. A number of people have been saying this. That there's the cancer has characteristics of immune cells, right? So and current Candace Pert, a very powerful, recognized scientist, said the same thing, and she was vilified. And the attack, she published it in Science, saying that lung cancer is a macrophage kind of a disease. And there were people that attacked her, and boy, I'll tell you, she laid into those guys like there was no tomorrow. I give her a lot of credit. She just tore the ass off some of these idiots that were challenging her work. She was the leader in the field, and you didn't look at the data. So it was great to see that happening. But
What's
yeah,
the mechanism at play here? Well, actually, first of all, you said Candace Pert?
Candace Pert, yes.
Okay, I'll look her up. But so the mechanism here is
Science, 1984.
okay.
Look at the paper, 1984 Science, with her husband, Ruff.
So the mechanism is that the white blood cells engulf a cancer cell, and then there's some kind of transformation of their own mitochondria that induces them to be cancerous as they go throughout the body, or like what is this?
Well, in a cancer situation, you have a lot of inflammation. You have a lot of necrosis. You have a lot of mess there. And our immune system is designed to heal wounds. This is what we do. So if I cut myself and lead a big contusion, I've got hypoxia, I've got bacteria there, and then my monocytes come pouring out of the bloodstream and differentiate into macrophages to facilitate wound healing with fibroblasts and also killing bacteria that might be in there, because it's going to be a race for life or death. If the bacteria win, you die from sepsis, you die from bacterial infection. If your immune system wins, you kill the bacteria, you heal the wound, you got a scar, and you survive. Cancer is like an unhealed wound. Cancer is growing, creating the same microenvironment as you would have in a wound. The problem is the cells that are creating the disturbed microenvironment are fermenters, but they can't metastasize. They might be like some stem cell thing. So they grow, but they're throwing out acidified lactic acid and succinic acid, signaling the immune system to come in. The immune system comes in and throws out cytokines and growth factors, thinking they're facilitating wound healing, but it's actually making the situation worse. It's doing something evolutionarily programmed in a wrong context. So, and it gets worse and worse. Now, macrophages have a unique capability. They can fuse with each other to facilitate wound healing, or they can fuse with other cells in the microenvironment. So if I have a bunch of stem cells with already corrupted oxidative phosphorylation and mitochondria, and they are fermenting, and the macrophage comes in and cytoplasmically fuses with the cytoplasm of the stem cell, all of a sudden you get a mixing and a dilution of the mitochondria and the macrophage. All of a sudden now you have a cell that can ferment, but now it's dysregulated because the organelle that's regulating that cell has now become part of the problem. So you have the most powerful cell in your body running rogue, but it has the genetic capability of suppressing normal immune cells, surviving in the circulation, called intravasation, where they leave the site, enter the bloodstream, go through the bloodstream, extravasate, and get into tissues spreading around the body, all because they are programmed to do this. The problem, the organelle that controls them doing it in the right way, is now corrupted. And as long as they have fermentable fuels, they're rogue macrophages, and that's the origin of metastatic cancer. I
I,
spent 10 years doing this.
I think it's really surprising. I mean, most people who have been through some college biology or even high school biology probably have heard of macrophages eating things, like
Yeah.
consuming, other cells, bacteria, things like that. But I don't think that the fusion mechanism is very widespread, that people understand that the macrophages will fuse with
Oh, yeah.
wounded tissue. I've never heard that before.
But I'm also struck by how kind of dumb my understanding of metastatic cancer was, because I'm going to expose myself as being a total idiot. I just figured they grew legs and just kind of walked away.
No, they're part of our immune system. Yeah, and you can kill them because we know what they eat, and that's why I tell you, first thing you do is, they're big. That's why I call them macrophage, big eater. Okay, so they eat stuff. So and that's another thing too. When they're starving, they will eat each other. That's why you really have to be. And they'll eat the microenvironment stuff, and they'll eat microbeans. We tested all this stuff, so I know what they are, and I know how to kill them. The problem is I tell people that, and they look at me like, "Oh, I never heard of that before." Well, for Christ's sake, you're not doing the research like we are. You're not reading the papers like we are. There's plenty of information on it.
Are you hopeful that people will just read your papers and attempt to institute this in their own lives?
Well, that's a hope. Yeah. I mean, obviously, if I didn't care about it, I wouldn't have published any of that. I just keep it to myself. So I would know if I got metastatic cancer, I know exactly what to do. And I feel bad for everybody else who doesn't know what I know. It's not.
I'm really excited to share this conversation with people. There's a bunch of people in my life that need to hear this, and I'm so grateful we got to finally sit down with you and lay it out. It's super, super important. I've had incredible changes in my own health as a result of some dietary modifications along these lines. Not cancer related, but I really do think that we have a lot more power than just going and getting some pills from the doctor. And this is super, super inspiring to me.
It's wildly empowering.
Yeah,
right?
well, that's
the idea that, I mean, if a death sentence is handed down to you by a doctor, the idea that there are things that you can do and help your own body survive is just magnificent.
And the thing you'd want is the person that would know this. So the team is the patient's knowledge and the physician's knowledge together, working together as a team. To work with that individual, this didn't work so well for you. Let's just try that. But we're still putting pressure in this direction. So in other words, you have two knowledgeable individuals, and the outcome will always be significantly better than if either of them don't know anything, or one knows and the other one doesn't. So you've got to have a team effort in managing cancer. But it also will manage all these different chronic diseases as well. And when you consider the enormous fund of how much money our country and other countries are spending on diabetes, cardiovascular disease, Alzheimer's disease, cancer, where all of these can be managed basically with the knowledge of keeping your GKI low and bringing your body back into some sort of a Neolithic period, and recognizing those products of our great technology that can be greatly enjoyed under certain conditions. So it's going to be a lot of knowledge, because we're going to evolve. Who we are today, if we live, may not be who we are in a million years from now. We're going to know a lot of things that are called mysteries and unknown will become known if we can survive as a species. But knowing facts and replicating facts are the basis for the development of theories. So once you have a significant observations that are repeated over and over again, you develop a theory to explain that phenomenon. And as you begin to develop theories to explain the phenomenon, we move forward in what we can do. And this was the same thing with the heliocentric geocentric theory. It was the Copernican revolution that said it was the sun, not the earth. So you could explain the movement of the planets. You could explain things. You could better understand the universe based on these kinds of things. And this is the same thing. Once you understand the biology of cancer and you put the pieces together, you can move forward with disease management and understanding ourselves better. So this is the future. It's just that right now you have a lot of obstacles that need to be knocked down in order to move the field forward. But it's going. It's coming.
I mean, it's the same in all disciplines. Like Shiloh and I, the purpose of this podcast is to work on lowering those barriers. And so we've actually, in addition to the podcast, we've started organizing conferences. And so this year we're doing one for astrophysics and cosmology, where we're bringing together, the orthodox thinkers, the philosophers, the observers that are looking through telescopes, and we're calling it Beyond the Big Bang. And so the scientists are going to collaborate to publish like a roadmap for cosmology in the aftermath of the conference. And so it's really, I think the future is creating a new scientific society where people actually have the ability to explore these sorts of topics from a position that is not ideologically or financially captured.
Yeah, well, this is the advance of science. This is the new revelations that you see, these paradigm changes. This is the way it works for sure. So anyway, I'll let you guys go. I have a meeting that I have to get to, but get yourself a keto mojo. Start measuring your GKI and finding out what zone you guys are in. So when you go to a cocktail party, it now becomes a point of discussion. Oh, how's your GKI today?
Oh no! Oh no!
We already have hard enough time at cocktail parties.
Well, whatever party you go to, whatever group you go to. But yeah, it's going to be a transformation, which is exciting.
Yeah.
Whenever you have a new generalization, it makes people think about things they previously didn't think about.
It's a hell of a time to be alive.
Yeah. All right. Well, thank you so much. We salute your efforts to the highest degree, and we're super excited about this work.
And let people know that if they want to support our research, they can support it at Boston College, because we depend on those folks, the philanthropists, the people who say, I don't really care if I make a buck on this. I just want to be part of the movement. And it's going to be exciting. So, or Travis Christofferson's foundation, these kinds of things, because that's how we keep going. So now after I have to go to this meeting, what I do is I have these same conversations with my group next door, and we talk about these things for hours and hours and hours, always looking for a fatal flaw in our concepts, and trying to identify any blind side that we might get from some piece of evidence that might be contradictory to what we think. And then we evaluate it and we do experiments to test it. So that's the way we.
I love it.
Good luck out there. Thank you so much.
Thank you.
Okay, guys. Have a nice day now.
You too.
Bye, everybody.
Bye.